Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Okay, imagine this for a second. You're told about a disease, right? Spreads like an infection. But here's the kicker. No DNA, no RNA involved. A protein. I mean, that idea completely messes with the central dogma of molecular biology we all learned. And it was the core proposal in this really groundbreaking 1982 paper in Science. So for this deep dive, our mission is to really get into that pivotal research. It's called Novel Proteinaceous Infectious Particles Cause Scrapie by Stanley Prusiner. And honestly, it didn't just add a little bit to what we knew. It totally reshaped how we think about infectious disease. It's huge.
Absolutely. And to kind of set the scene for Prusiner's really bold idea, let's talk about scrapie itself. So for decades, this disease in sheep, scrapie, is fatal neurodegenerative. It was just a puzzle, a real biological enigma. People thought maybe it was some kind of unconventional slow virus because, you could transmit it, that part fit. But the agent, the supposed virus, acted so strangely, really weirdly. It resisted things that should destroy nucleic acids, right? Like radiation, enzymes that chop up DNA and RNA. But it was sensitive to stuff that messes up proteins, so it just didn't fit the mold at all.
Okay, so hold on. You've got this infectious thing, causes disease, but it shrugs off treatments that should kill any normal virus by destroying its genes. But protein-damaging stuff does hurt it. That's baffling, given how deeply everyone believed that, genes' DNA or RNA had to carry the infectious blueprint. What was the leap Prusiner's group made? How did they even start thinking differently? It must have seemed way out there.
Oh, absolutely, way out there. What's so fascinating is that Prusiner and his team looked at all this data and said, okay, this scrapie agent, it just doesn't meet the criteria for a virus, period. It defies the rules requiring nucleic acids. So they took this incredibly radical step. They hypothesized a totally different kind of infectious thing, an abnormal protein, just the protein. And their goal wasn't small, it was audacious. They aimed to prove, experimentally, a direct causal link between a specific protein and a transmissible disease. That meant directly challenging that bedrock dogma, that hereditary information must live in nucleic acids. And yes, you're right, it was massively French. The skepticism was immense.
Wow. Okay, that context makes what they did even more impressive. So this radical protein-only idea, how did they even start building a case? What were the first key experiments, the first bits of evidence that started to maybe, just maybe, make people reconsider?
Well, they started by being incredibly meticulous. They took brains from hamsters infected with scrapie and started purifying the infectious agent from them. And one of their first really key experiments, you think of as like figure 1 in their story, involved treating these purified preps with something called proteinase K. It's basically a molecular shredder for proteins, very effective. And what they saw was, well, remarkable. As the proteinase K destroyed the proteins in the sample, the infectivity also disappeared. It went down right alongside the protein degradation.
Ah, okay. So destroying the protein destroyed the ability to cause infection.
Exactly, and that was a huge clue, because if it were a normal virus, damaging its protein coat might weaken it, sure, but as long as its DNA or RNA was intact, it should still be infectious.
Right, the genetic material should still work.
Precisely. So this finding strongly pointed towards a protein being essential for the infectivity itself. And they even identified a specific size range. A 27 to 30 kilodalton protein seemed to be the key player.
Okay, that's pretty compelling for the protein angle right from the start, but I can still hear the critics, Okay, fine, a protein's involved, but where's the nucleic acid? It has to be there somewhere. How did they tackle that? How did they try to rule out DNA or RNA definitively? Like, what was figure 2?
Exactly the question everyone was asking. So next, they basically threw the kitchen sink at the scrapie agent, but only things known to destroy nucleic acids. They used intense UV irradiation, which scrambles DNA and RNA. They used cocktails of nucleases, those enzymes you mentioned that specifically chew up nucleic acids. They used sorlin cross-linking, which chemically binds up DNA and RNA so they can't function.
Okay, so basically a death sentence for any known virus or anything relying on genetics.
Absolutely lethal. And the crucial finding, none of these treatments inactivated the scrapie agent. Not one bit.
Wait, really? Infectivity remained intact after all that?
Completely intact. It was just as infectious as before, and that directly contradicted the whole idea of a conventional virus being involved. It was incredibly powerful evidence against the necessary nucleic acid component.
Okay, wow. That seems hard to argue with. No nucleic acid needed. But how did they zero in on which protein it was? Was it just any protein or that specific 2730 kiloday one they saw earlier? How did they connect the dots?
All right, that was the next logical step, wasn't it? They had to show it wasn't just a protein, but that protein.
Right.
So they focused all their efforts on enriching or purifying that specific 2730 kiloday protein. And they're really good at it. Using techniques like differential centrifugation, detergent extraction, basically ways to separate molecules based on size and properties, they managed to purify this protein fraction over 100,000-fold.
100,000 times pure. That's intense.
It's seriously pure. And here's the clincher, this sort of final figure moment. They found that the level of infectivity always tracked perfectly with the concentration of that specific protein. Wherever they found more of that 2730 kiloday protein in their purification steps, they found more infectivity. They co-fractionated, meaning they stayed together throughout the whole process.
So they were essentially inseparable. The protein was the infectiousness.
That's what the evidence strongly suggested. And just to be thorough, they did spectral analysis on their most purified samples.
Yeah.
Looking for that characteristic absorbance peak that nucleic acids have.
Yeah.
Nothing. No peak. It basically confirmed there were no detectable nucleic acids left.
So step by step, experiment by experiment, they built this incredibly robust case pointing squarely at a protein. Just a protein causing this disease. That must have been, well, mind-blowing, but probably really hard for people to swallow.
Oh, absolutely. It flew in the face of decades of biological understanding.
So what did they end up calling this completely new kind of infectious particle?
Well, given that it was so clearly different, not a virus, not a plasmid, not these other things called viroids, Prusiner needed a new name. He proposed the term prion. It's actually a portmanteau, a blend of words. It stands for a small proteinaceous infectious particle.
Mm-hmm.
And the key part of the definition was that it's resistant to inactivation by most procedures that modify nucleic acids.
Prion. Huh. So proteinaceous infectious particle, it really spells it out. This wasn't just like another paper adding data points, was it? This sounds like a conceptual earthquake.
Yeah.
How exactly was the research innovative beyond just the shocking finding itself?
It was innovative on multiple levels, really. Conceptually, obviously, it provided the first real molecular evidence for an infectious agent made only of protein. That alone birthed the whole field of prion biology and directly challenged that central dogma we keep mentioning. But methodologically, it was also groundbreaking. They essentially pioneered this technique of tracking infectivity across all the steps of biochemical purification. It allowed them to rigorously correlate the disease-causing ability, the pathogenicity, with a specific molecule, in this case, the prion protein. It wasn't just what they found, but how they systematically proved the connection. That became a template for other research.
Right. It's like they provided a roadmap for how to track down unconventional infectious agents. That makes sense. And what about the people involved? You mentioned Prusiner, but big discoveries usually involve a team, right? Especially when facing so much skepticism.
Oh, definitely.
Yeah.
The team at UCSF was absolutely critical. Stanley Prusiner, of course, he provided that initial high-risk conceptual leap. Yeah. And he really had to push, to persevere against widespread doubt, even ridicule sometimes. But then there was his postdoc, Patrick Bolton. He was apparently instrumental in developing that key proteinase K treatment protocol and meticulously quantifying the infectivity, linking it to the protein degradation. That's painstaking work. And you also have to mention the technicians, like Diane McKinley. She managed the incredibly demanding work of serially passaging the disease in hamsters, infecting one group, waiting, harvesting tissue, preparing inoculum, infecting the next.
Oh, wow, yeah. That sounds like a huge amount of careful, repetitive work needed for statistical power.
Exactly. It ensures the results aren't just a fluke. So their collective persistence, their meticulous lab work, all while people were saying this can't be right, that was absolutely fundamental.
It really highlights the human effort behind the science. So fast forward from 1982, what does this discovery mean for us today? Where did this research lead? What are the applications?
Well, connecting it to the bigger picture, the implications turned out to be enormous. Identifying the abnormal prion protein, which they later called PRPSC, was the crucial first step towards developing diagnostic tests. And those tests became incredibly important for public health, especially during the whole bovine spongiform encephalopathy, mad cow disease crisis in the UK and elsewhere. being able to detect the agent was vital.
Right, for tracking the disease in cattle and preventing human transmission.
Precisely. But maybe even more profoundly, the whole concept that a misfolded protein could somehow induce other proteins to misfold and propagate like an infection,
Yeah.
that idea now underlies research into many other major neurodegenerative diseases. We're talking about conditions like Alzheimer's and Parkinson's. There's growing evidence that prion-like mechanisms, where proteins misfold and spread through the brain, play a key role in those diseases too. So the assays developed based on Prusiner's work are relevant there as well.
That's incredible. So a study on a weird sheep disease basically opened up entirely new ways of thinking about some of the most devastating human brain diseases. And even though the original paper maybe didn't have all the answers back then, like maybe they hadn't purified the protein to absolute homogeneity or fully explained how different prion strains could exist without genes, it was clearly a massive paradigm shift.
Undeniably. It launched a whole new field focused on protein misfolding diseases. And of course, it eventually led to Prusiner winning the Nobel Prize in 1997. It was a hard-won validation.
Looking back at it all, what do you think was the single biggest hurdle for the scientific community to actually accept this prion concept? Was it just the lack of Dino or Nana?
Yeah, I think fundamentally it was that challenge to the central dogma. For decades, biology was built on the idea that information flows from nucleic acids to proteins, and that replication requires a nucleic acid template. The idea that a protein all by itself could somehow contain infectious information and replicate by corrupting other proteins. It just sounded wrong. It almost felt like it violated basic principles. It took many more years of really detailed biochemical and genetic work figuring out the normal PRP protein and how the abnormal PRP C form converts it for the concept to become widely accepted. The mechanism had to be plausible.
So reflecting on this deep dive, what really stands out to you? For me, it's just the sheer audacity of it. Challenging something so fundamental, not with just an idea, but with rigorous step-by-step experimental evidence, even when everyone else doubts you. That protein-only idea was truly revolutionary.
I agree. And what this deep dive really underscores for me is how science progresses. Sometimes it's not about fitting new data into old theories. Sometimes the data, especially the weird, unexpected data from things like the scrapie agent, forces you to break the old boxes entirely. It's a reminder that even our most established principles aren't dogma forever. They're models, waiting to be refined or even overturned by persistent questioning and evidence that just refuses to fit.
Absolutely. And it leaves us with a really fascinating thought to chew on, doesn't it? If just a simple protein, misfolding, can act like an infectious agent and cause such devastating neurodegeneration, what other complex processes in our bodies, maybe even perfectly healthy ones, might be governed not just by our genes, but by this subtle, intricate dance of protein shapes, conformations, and interactions propagating information in ways we're only beginning to understand?